However, because you have already done this, I will not lecture to you anymore the digestion and absorption part. So we go direct to protein turnover. Now, what do you mean by protein turnover? This is the balance between proteins undergoing breakdown or catabolism or degradation and the proteins that are being synthesized every day. So araw-araw yan. Now, about 1 to 2% of your total body proteins undergoes protein turnover. In terms of grams, that will be 200 to 300 grams every day. So 200 to 300 grams undergoes catabolism, 200 to 300 grams undergoes synthesis also every day. So your proteins are not dormant. All right? Now, when proteins undergo breakdown, of course, the final end product are your amino acids. 75 to 80% of this will be reutilized, recycled. You're going to use them again for protein synthesis. 20 to 25% are converted to urea. Urea represents the final end product of protein metabolism in your body. Final end product of protein metabolism. And this is excreted in your urine. So, the amount of urea that you excrete in your urine is directly proportional to your protein intake. If your protein intake is high, if you eat a lot of proteins, you expect the urea excretion to go up. But if your protein intake is low, naturally, your urea excretion will be down also. The susceptibility of your proteins to degradation is expressed as its half-life, or how long does a protein stay before it is degraded. And there are, if you want to know the full life, you just multiply it by 2, the half-life by 2. There are proteins whose half-lives are below 30 minutes. So that means the protein does not even stay more than 1 hour in your body. It undergoes degradation already. So this is referred to as short half-life, less than 30 minutes. But there are proteins with long half-lives, over 150 hours. 150 hours in terms of days, that will be around 6 to 7 days. Multiply it by 2. So the protein stays for about 2 weeks inside your body before it undergoes degradation. So that is referred to as long half-life. Now, proteins with short half-lives usually have the PEST sequence. Remember, in the formation of your proteins, you connect the amino acids to one another through the peptide bond. So you have a definite sequence of amino acids in the peptide chain, starting from the N-terminal. N-terminal refers to the first amino acid in the peptide chain, down to the C-terminal, the last amino acid in the peptide chain. Now, if you recall, when you had your lecture in protein chemistry, you were told that there are 2 types of abbreviations for amino acids. The one that you use is the 3-lettered abbreviation. It's easier to remember the 3-lettered abbreviation because it's usually the first 3 letters. So alanine is ALA, your valine is VAL, your phenylalanine is PHE, but there are some exceptions to the rule, like isoleucine. It's not ISO, but ILE. Tryptophan is not TRY, but TRP. So those are exceptions to that first 3 letters. Now, there is a 1-lettered abbreviation, and this is not usually the first letter in the name of the amino acid. For example, for the PES sequence, the P is for proline. But E is for glutamic acid, not G. S for serine, T for threonine. Okay. So if you find the sequence of amino acids in a peptide chain, then it has a short half-life because it is a target for rapid degradation. Now, proteins with half-lives over 100 hours, long half-lives. This includes aldolase. In what pathway did you meet aldolase? Answer? All right, very good. This is the enzyme called cleavage enzyme in glycolysis. It cleaves fructose 1,6 bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, of course, lactate dehydrogenase is the enzyme that converts pyruvate to lactate when the condition becomes anaerobic. And cytochromes are members of your electron transport chain. Now, the most rapidly degraded enzymes, remember enzymes are proteins. So when you degrade them, you also get amino acids. The most rapidly degraded enzymes are those that occupy important metabolic control points. What do you mean by this? The more important the function of the enzyme is, the rapid is its degradation. So you expect that all the rate limiting enzymes or the so-called committed enzymes or the major control enzymes, they are most rapidly degraded. Okay. So if you compare the enzyme aconitase versus the enzyme citrate synthase, which one will be more rapidly degraded? Aconitase or your citrate synthase?
Citrate synthase.
Citrate synthase.
Citrate synthase, because citrate synthase is the rate limiting enzyme of the Krebs cycle. And aconitase is just an ordinary enzyme. And if you're going to compare your glucose 6-phosphate dehydrogenase versus your phosphoglycerate kinase, which one is more rapidly degraded? All right. Glucose 6-phosphate dehydrogenase, because that is the rate limiting enzyme of the HMP shunt. And your phosphoglycerate kinase is just a reversible enzyme of glycolysis. Remember, that's the enzyme catalyzing the first substrate level phosphorylation in glycolysis. So you must remember all the rate limiting enzymes. For glycolysis, what is the rate limiting enzyme?
Phosphofructokinase 1.
All right. Phosphofructokinase 1. And for glycogenesis, what is that control enzyme there? Glycogenesis.
Glycogen synthase.
Okay, glycogen synthase. The opposite of glycogenesis is glycogenolysis. What's the control enzyme?
Glycogen phosphorylase.
Glycogen phosphorylase. Very good. Now, when it comes to the HMP shunt, G6PD, as I've said, is the rate limiting enzyme. Although transketolase and transaldolase are also control enzymes. Pero mas rate limiting si G6PD. So the more important the function of the enzyme is, the more rapidly it undergoes degradation. Also, the rate of protein degradation depends upon your nutritional and hormonal status. If you are nutritionally deprived, you are malnourished, you are starving, there will be a more rapid protein degradation. Why? If you recall the lecture on carbometab, ako nag-lecture, di ba? Carbometab?
Yes po.
Yes
Oh, okay. Tama. Kasi wala namang, usually, I remember, Kasi there was a year when I was not able to lecture carbometab because that was the time that I went to the US. I had my trip, US-Canada trip. So that was 2 months. I was out of FEU then. So Dr. Alcantara lectured. Pero because of the COVID, no traveling. Okay. So I was the one who lectured. So I told you that what maintains every day your blood sugar level? What? Why don't you go into hypoglycemic shock every now and then? What maintains your normal blood sugar level? You forgot. The diet. Diba? You eat every day. The carbohydrates in your diet maintains your blood sugar level every day. 2 hours after you eat, your blood sugar tends to go down. That's why you feel hungry again. But if your blood sugar tends to go down, you stimulate your glucagon. Your glucagon will do glycogenolysis or gluconeogenesis now to pull that blood sugar back to normal. So it is the diet that maintains your blood sugar level every day. But when you cannot eat, for some reason or another, you have nothing to eat or you cannot eat because you are on top of your house, there is a big flood in your subdivision. You are now on top of your house. You were not able to bring any food. So, it's almost 12 hours already. You haven't eaten. Why will you not go into hypoglycemic shock? What will you now use to maintain your blood sugar?
Glycogen.
Okay, glycogen. Very good. Glycogenolysis. That's breakdown of glycogen to glucose that will now maintain your blood sugar so that you do not go into hypoglycemia. But your glycogen can only sustain your blood sugar for at the most 18 hours. So if you still cannot eat after 18 hours, what will you now use so that your blood sugar will not go down?
Fats po.
Your fats. Your fats will undergo breakdown to fatty acids and glycerol. And it is the glycerol part that will undergo gluconeogenesis. Glycerol, it's not the fatty acid that is converted to glucose. Now, for how long does your fat sustain you, the glycerol sustain you? Depends upon how much fat deposits you have. So this is one advantage if you are more plump, mas mataba ka, because you will have more fat deposits to break down the fatty acid and glycerol, which can become glucose. So that can even sustain your blood sugar level for 1 week. So you will not die even if you do not eat for 1 week, for as long as you have water and air, of course. Cannot be if you don't have water and air. So, if all your fat deposits have been used up, what now? You still cannot eat, you still do not have food to eat, no more fats. You will now use your proteins. Your muscle proteins undergoes degradation to release your amino acids. Amino acids now will enter gluconeogenesis to become glucose. So, if your nutritional status is not good, there is a more rapid degradation of your proteins. Kasi sila kagad yung magagamit mo eh. Mauubos kagad. Your glycogen, your fats will all be used up. And so, you now have to use up your muscle proteins. Okay. Now, there are two pathways responsible for the degradation of proteins. One is your lysosome. Your lysosomes preferably degrades proteins containing the pentapeptide, 5 amino acids, KFERQ. Now, as I've said, the one-lettered abbreviation do not usually use the first letter. Here, the K stands for lysine F for phenylalanine, E for glutamic, R for arginine, and Q for glutamine. Ibang-iba. That's why it's harder to remember the one-lettered abbreviation. But if you have to take the US exams, you have to know the one-lettered abbreviation because they use the one-lettered and not the three-lettered abbreviation. So lysosomes preferably degrades proteins containing the pentapeptide KFERQ. The other pathway is your ubiquitin. This also prefers to degrade peptide chains where the N terminal or the amino terminal or the first amino acid is aspartic or arginine. And they cannot degrade if the N terminal is methionine or serine. So specific din yan. Okay? Now, this is the overall scheme for protein metabolism. So your amino acids actually comes from two sources. One, exogenous source, outside. So this is your diet. How many grams of protein should you eat per day? Are you done with nutrition lecture? What's your daily requirement for proteins? This is 1 gram per kilogram of your ideal body weight. Now take note, your ideal body weight is not always your actual weight. More often than not, the actual weight is more than the ideal body weight. How do you get the ideal body weight? Based on the Tannhauser's formula. You have to know your height. So if your height is 5 feet tall, convert that first to centimeters. 30 centimeters per foot. So 30 times 5, 150. Minus 100. You get 50 kilos. That's the ideal body weight based on Tannhauser's. Now for Filipinos, we still deduct 10% of that 50 because you have to consider that the build of Filipinos is smaller compared to Americans. So 10% of 50 is 5. 50 minus 5, your ideal body weight, if you are 5 feet tall, is 45 kilos. That's more or less 90 to 92 pounds only. No? So, 1 gram per kilogram of ideal body weight. You should eat 45 grams of protein per day. Okay. Now, it is written there 70 grams. This is for Americans. Just remember the textbook is American. So that's why approximately 70 grams. Aside from the exogenous source, you have the endogenous source inside. Now, this refers to the amino acids that are synthesized inside the liver. And these are your non-essential amino acids. This will also include the amino acids coming from the breakdown of your tissue proteins, the turnover amino acids. So those are the sources. Now, where do you use amino acids? Of course, number one use of amino acids in your body, you use them for protein synthesis. So you synthesize your hemoglobin, your myoglobin, your antibodies, collagen, keratin. Remember, collagen is the most abundant protein in your body. And enzymes are also synthesized. Remember, enzymes are proteins. Now, you also use amino acids to synthesize the so-called essential non-protein nitrogenous substances. These are substances that are not proteins, but you need amino acids when you synthesize them. Example, purines and pyrimidines, when you synthesize them, you need to have amino acids. For the purine ring, nitrogen number 1 of the purine ring comes from aspartic acid. Nitrogen 3 and nitrogen 9 and 3 and 9 comes from glutamine. And carbon 4, carbon 5, and nitrogen 7, C4, C5, and 7 comes from glycine. So, in the synthesis of your purines, you need to have aspartic, glutamine, and glycine. Okay? Now, in the synthesis of porphyrins, you're done with this. What's the amino acid that you need to synthesize your porphyrins?
Glycine po, Doc.
Glycine. Very good. Glycine plus succinyl-CoA are the precursors for heme. Heme is a porphyrin. Also, there are some vitamins that contain amino acids in their structure. Example is folic acid or vitamin B9. It contains glutamic acid in its structure. Creatine is a tripeptide. It has 3 amino acids: GAM, glycine, arginine, methionine, GAM, creatine. Another important tripeptide is glutathione. Glutathione has GCG, glycine, cysteine, glutamic acid. All of these are not proteins, but you need amino acids for their synthesis. If any amino acid is not used, it will undergo catabolism or breakdown. You do not store amino acids. Unlike carbohydrates, which you store as glycogen, and fats, which you store as triacylglycerol, the amino acids will not be stored. You will just catabolize them, or they will undergo degradation. And these are the different ways by which amino acids can be catabolized: transamination, deamination, decarboxylation, etc. We will discuss that in detail. So that's the overall scheme for your protein metabolism. Now, after digestion and absorption, where will the amino acids go? They go to the liver. The liver plays a central role in the metabolism of amino acids, not only amino acids, but also carbohydrates and lipids. So from the point of view of nutrition, the most important organ in your body is the liver. Without the liver, you cannot metabolize anything that you eat. So, it is very easy to die if you cannot get the nutrients out of the foods that you eat. You may eat them, but you cannot metabolize them, so you do not get the necessary nutrients from the foods that you eat. That's why it is so easy to die if you have a liver disease. In 1 to 2 months' time, you can die. If you happen to have a liver CA, hepatocarcinoma, liver cancer, 1 month, 2 months at the most, even with treatment, they die. Mabilis. Very, very fast. One liver disease that you must be careful of is hepatitis B. In contrast to A, hepatitis A is treatable. But hepatitis B is not. That's why be sure that before you go to second year, when it's face-to-face, you have your hepatitis B vaccine, because you will be handling patients in a subject called physical diagnosis. You're going to interview patients. You're going to practice getting history taking. So you will be interviewing patients. And hepatitis B can be transmitted by droplet infections. So it's not treatable yet. So you should have your hepatitis B vaccine before you go to your second year. We used to have research where we give the vaccine to the first year students because we were comparing the efficacy of the two vaccines. The DNA-derived vaccine is Engerix, and then we have the plasma-derived vaccine, Hepaxine. These are the two kinds of Hep B vaccines that are being given. So half of the students were given Engerix, half were given Hepaxine. Now, both of them are effective, kaya lang, for Engerix, the protection is longer, which means if you had Engerix, your booster dose could be given after 5 years. But if you took Hepaxine, your booster dose should be given after 1 year already, because there is the tendency for the antibody level to go down for Hepaxine very early. So see to it that if you have your Hep B vaccine, mas okay si Engerix. Okay? So what happens in the liver? All amino acids are metabolized in the liver except 3. What are the exceptions? The branched chain amino acids: valine, leucine, isoleucine. They are not metabolized in the liver. They're metabolized in the brain and in the muscles. All the rest, they're metabolized in the liver. So what happens in the liver? As mentioned a while ago, you synthesize the proteins that your body needs. Enzymes, immunoglobulins, collagen, keratin, hemoglobin, etc. You synthesize the non-protein nitrogenous substances, purines, pyrimidines, porphyrins, etc. The liver also delivers amino acids to other organs, so it is a central supplier of amino acids. It is also in the liver where you synthesize the so-called non-essential amino acids. We will discuss that later. Now, if the amino acid is not used, it is going to be catabolized. It is not going to be stored. It will just undergo catabolism. Now, let's talk about the classification. In protein chem, you had a lot of classifications. You can classify them whether they are neutral, acidic, or basic. You can also classify them according to their functions. You can have your transport proteins, regulatory proteins, things like that. But you can also classify them according to their nutritional status. So from a nutritional point of view, you have the so-called essential or indispensable amino acids. These are amino acids that cannot be synthesized by the body. So you must provide it in the diet. Kailangan kainin mo because your body cannot produce it. You must eat it. 10 of them. Now, if all these 10 essential amino acids are found in a protein, the protein is called complete protein, or it is a protein of high biological value. And a complete protein sustains life by itself. Pwede kang mabuhay na yun lang kinakain mo. You can live even if that is the only one that you are eating. What are the complete proteins? Protein found in milk is complete. That's why babies live and grow even if they drink milk alone. A newborn baby does not eat anything except milk, but he grows, he lives, because the protein that is found in milk is complete. and can sustain life by itself. Other sources of complete protein are meat and eggs. Milk, meat, and eggs. Now, if anyone, just one of these essential amino acids is lacking, the protein now becomes incomplete, or it is a protein of low biological value, even if there is only one that is lacking. Examples of incomplete proteins are those that are found in cereals, fruits, vegetables. Rice has a protein. Do not think that rice is pure carbohydrates. There is a protein in rice called oryzanin. Oryzanin. But it is an incomplete protein. Likewise, corn has zein. Zein. That's the protein of corn. It is also incomplete because it lacks tryptophan only. Now, the amino acid that is lacking in an incomplete protein is called limiting amino acid. So in zein, the limiting amino acid is tryptophan. Non-essential or dispensable amino acids are those that your body can produce. You can synthesize it. So there's no need to provide it in the diet. No need to eat it because you can produce it anyway. There is one amino acid that is referred to as semi-essential. Semi-essential. It can be synthesized by the body. So it should be non-essential because you can synthesize it. However, you must provide it also in the diet. So it becomes essential. So it satisfies both definitions. That refers to arginine. It is semi-essential because you can synthesize it through the urea cycle. So that's the non-essential aspect. But you must provide arginine in the diet if you want to use it for protein synthesis. So that satisfies essential. So it is both essential and non-essential. That's why it is semi-essential. You will know later why you must provide arginine in the diet even if you can produce it when we discuss the urea cycle. All right. Now, for the synthesis of the non-essential amino acids, there are three important enzymes that can be used. Glutamate dehydrogenase. This is for the synthesis of glutamic acid alone. Glutamine synthetase is for the synthesis of glutamine alone. Aminotransferases, you also call this transaminases. These are enzymes that are used for transamination, and they are used to synthesize many other amino acids. So what's the difference of the three enzymes? Glutamate dehydrogenase is for glutamic only. Glutamine synthetase is for glutamine only. But aminotransferases can be used to synthesize many other amino acids. You can synthesize alanine, aspartic acid, glutamic acid using aminotransferases. So, hindi lang iisa yun. Now, when an amino acid undergoes catabolism, what happens? If you recall, what are the three active parts of an essential of an amino acid? You have the alpha amino group, you have the alpha carboxyl group, They are both attached to the alpha carbon. And of course, the R group. Those are the three reactive parts: amino, carboxyl, R group. So if you want to catabolize or break down an amino acid, you just remove one by one the three reactive parts. Tanggalan mo lang isa-isa yan. When you remove the R group, that's called one carbon transfer. Remove the R group. Siyempre, if you remove the R group, the amino acid is no longer an amino acid. Or you can do oxygenation. You are not removing anything. You just have to add oxygen. Example of oxygenation: when you add oxygen to phenylalanine, it is converted to tyrosine. That's still considered to be catabolism because you are converting phenylalanine to another substance. Decarboxylation, this is removal of the carboxyl group, the COOH group, and this is converted to carbon dioxide, CO2. Now, the remaining part of the amino acid, once you remove the carboxyl group, is now called an amine. Amine. Example: when you remove the carboxyl group of histidine, it becomes histamine. When you remove the carboxyl group of tryptophan, it becomes serotonin. Phenylalanine and tyrosine becomes catecholamines, norepinephrine, and epinephrine. So may kanya-kanyang amine, bawat amino acid. For the amino group, you can remove it through oxidative deamination, non-oxidative, or transamination. When you do oxidative deamination, this is the general equation: amino acid minus the amino group. You will have ammonia. The NH2 is converted to NH3. Then the amino acid that remains after you remove the amino group is called a keto acid. Example: if this is glutamic acid, the keto acid that you get is alpha ketoglutarate. If this is aspartic acid, you get oxaloacetate. If this is alanine, you get pyruvate. If this is glycine, you get glycolic acid. If this is histidine, you get urocanic acid. So each amino acid has its corresponding keto acid. Now this reaction can be reversed. You can combine again the ammonia and the keto acid. You get back the amino acid. So ammonia plus pyruvate, you get back alanine. Ammonia plus oxaloacetate, you get back aspartic acid, and so on. Glutamate dehydrogenase is the most active enzyme for oxidative deamination. As I mentioned, this is just for glutamic acid. It converts glutamic acid to alpha ketoglutarate. Now, this is the most active enzyme for oxidative deamination. It is found in all parts of your body, even in the brain. And because it is the most active enzyme, it follows that glutamic acid is the most active amino acid. When you reverse, if this is alpha ketoglutarate, and you add again an amino group, then you call alpha ketoglutarate the most active amino group acceptor. It will accept again the amino group to become glutamic acid again. So the most active amino group acceptor is alpha ketoglutarate. The most active amino acid is glutamic acid. And the most active enzyme for oxidative deamination is your glutamate dehydrogenase, which is found in all parts of your body. Another enzyme for oxidative deamination is the L-amino acid oxidase. This will now act on the other amino acids because glutamate dehydrogenase is just for glutamic acid. Now, there is a D-amino acid oxidase, but we do not use it. Remember, in the human body, the amino acids that you use for protein synthesis, they all belong to the L series, not to the D. You only have two D amino acids, dalawa lang. D-aspartate and D-serine. Now, of course, you have an L-aspartate and an L-serine, but D-aspartate and D-serine are two amino acids which have been found to be present. The amino acids aspartate and D-serine are present in your brain. So we do not use D amino acid oxidase. Dalawa lang yan eh. There is a syndrome where L-amino acid oxidase is deficient. You call this Zellweger syndrome. And this is also called cerebro-hepato-renal syndrome, which indicates the involvement of the brain, the liver, and the kidneys. So you have abnormal neural development, excretion of amino acids in the urine, amino aciduria, and the absence of peroxisomes in the liver. This is deficiency of L amino acid oxidase. For non-oxidative deamination, the same equation as oxidative. Amino acid minus the amino group, you get keto acid plus ammonia. Iba lang yung enzymes. So the enzymes involved, you have amino acid dehydrase. This will only act on the hydroxyl containing amino acids. These are amino acids with an OH at the R position. Serine, threonine, tyrosine. Now take note, this requires pyridoxal phosphate as coenzyme. Pyridoxal phosphate or B6 phosphate. You will notice as you study the different pathways of protein metabolism, majority of these reactions requires vitamin B6 as coenzyme. And the most active coenzyme form of B6 is pyridoxal phosphate. This is the reason why vitamin B6 is referred to as the amino acid metabolism vitamin. Majority of its functions are related to protein metabolism. Now, for carbohydrate metabolism, the one that is usually involved as coenzyme is thiamine, B1. And the most active coenzyme form of thiamine is TPP, thiamine pyrophosphate. So, the requirement for B6 depends upon how much protein you eat. If you eat a lot of protein, then your requirement for B6 also goes up because you need it for as coenzyme in the different protein pathways. Your requirement for B1 or thiamine goes up if your carbohydrate intake is high because you need the thiamine pyrophosphate as coenzyme for the different carbo pathways. How about lipids? What is that vitamin that is related to the requirement for fats? If you are going to recall, tapos na kayo maglipid metab, fats can undergo oxidative degradation to form fatty acid peroxides. Upon further oxidation, fatty acid peroxides are converted to radicals. Radicals are toxic. Radicals are carcinogenic. So if you follow the equation, fats upon oxidation becomes fatty acid peroxides. Further oxidation, they become radicals. So fats can predispose you to cancer. It has been found out through studies that people who are fond of eating fatty foods have 20 to 25% more chances of developing cancer than somebody who is not fond of eating fatty foods. Take note of that. So, you get radicals from fats. You must take large doses of vitamin E, because vitamin E is called a radical scavenger. It will convert the radicals to non-toxic substances, thereby preventing cancer. That is why vitamin E is an anti-cancer vitamin. It protects your fats against oxidative destruction, so you do not get radicals out of them. You do not get the cancer. So your requirement for your fats, requirement for your fats, for your vitamin E depends upon how much fat you are going to eat. More fats, more vitamin E to handle the radicals. Okay. Another enzyme for non-oxidative deamination is your amino acid desulfhydrase. This one acts on the sulfur-containing amino acids, cysteine, methionine. And just like your dehydrase, it also requires pyridoxal phosphate as coenzyme. Now, for transamination, as the name implies, you transfer the amino group. So when you say you transfer, there must be an acceptor of the amino group. So if this is the amino acid containing the amino group, the acceptor is the keto acid. Remember, a keto acid has no amino group. Siya yung tinanggalan mo ng amino group. So when you transfer this amino group of this amino acid to this keto acid now will become a new amino acid. And the amino acid where you got the amino group will now become a keto acid. So every time you are doing transamination, you are producing a new keto acid and a new amino acid. Example, if the amino acid is alanine and the keto acid is alpha ketoglutarate, the new amino acid now is glutamic acid. Because when you add an amino group to alpha ketoglutarate, it becomes glutamic acid. Alanine, when you remove its amino group, will now become pyruvate. That's the keto acid. So you can synthesize glutamic acid there. Now, if you change the keto acid to oxaloacetate, the new amino acid now will be aspartic acid. If you change again the keto acid to glyoxylic acid, the new amino acid now will be glycine. So iba-iba. That's why I said a while ago, if you are using glutamate dehydrogenase, that's only for glutamic acid. If you are using glutamine synthetase, that's only for glutamine. But if you are using aminotransferases, you can synthesize many other amino acids. Depending upon what keto acid you are going to use to accept the amino group from the amino acid. If you are using pyruvate, the new amino acid is alanine. If you are using alpha-ketoglutarate, the new amino acid is glutamic acid. If you are using glyoxylic acid, the new amino acid is glycine, and so on and so forth. That's why iba-iba. You can synthesize many other amino acids using your transaminases. Now, what is the major difference between transamination and oxidative, non-oxidative deamination? As I've told you, the equation for oxidative and non-oxidative deamination is amino acid minus the amino group equals keto acid plus ammonia. Compare that to transamination. Anong wala? In transamination, you do not release ammonia because the ammonia that you remove from the amino acid is immediately incorporated into the keto acid. So you do not release it as free ammonia. Whereas in oxidative and non-oxidative deamination, you release free ammonia. And when you accumulate ammonia, that is toxic. Toxic yan. So, your body prefers to do transamination because that will not release ammonia. Remember, if your ammonia becomes accumulated, it can lead to ammonia intoxication. And how is this manifested? Initially, you are going to have nausea, vomiting, headache. Initial. As more and more ammonia accumulates, you will now have blurring of vision, slurred speech. Nauutal ka na, lumalabo pa yung paningin mo. As more and more ammonia accumulates, you will now go into convulsive seizures. And then, you become unconscious. And then, you become comatose. And then, you die. Exactly that is what happens in ammonia intoxication. That happens if you have a severe liver disease. Because the liver is the major organ for the detoxification of ammonia. So pag disease yung liver mo, ammonia is not detoxified, it is going to accumulate, causing ammonia intoxication. So the body prefers to do transamination. However, there are amino acids that do not undergo transamination. Lysine, threonine, proline, hydroxyproline. They do not undergo transamination. Okay. So, you can have, tapos na tayo ng decarboxylation. You can have ammonia accumulating, and so your body is equipped with fighting mechanisms. Your body will try to fight. It will try to detoxify ammonia. And one way to detoxify ammonia is to reverse the glutamate dehydrogenase reaction. Reverse pabalik. If you are asked, what is the product of the glutamate dehydrogenase reaction? Answer, alpha-ketoglutarate plus ammonia. If you are asked, what is the product of reverse glutamate dehydrogenase reaction? Answer, glutamic acid. Magkaiba. So, when you reverse, you are using the ammonia to convert the alpha-ketoglutarate to glutamic acid. That's one way to lower the level of the ammonia because you are using it. Now, in the brain, among all the organs in your body, the brain is the most sensitive to ammonia. It's very sensitive. You can go into abnormalities of your brain if the ammonia goes up, But your brain has a fighting mechanism by itself, and that's glutamine formation. Whatever ammonia reaches your brain, the brain will try to detoxify it. Combine it first with glutamic acid to form glutamine using the enzyme glutamine synthetase. And then the glutamine that is formed is brought to the kidneys. Once in the kidneys, the reaction is reversed. Glutamine is converted back to glutamic and ammonia using another enzyme, not glutamine synthetase. The enzyme that will convert the glutamine back to glutamic acid and ammonia is called glutaminase. Okay? So glutaminase. Now, that is in the kidneys. So, whatever ammonia is released, you can now excrete it through the urine. So, out goes the ammonia. The level goes down kasi lumabas na siya sa urine. So, if you are asked, what is the major source of ammonia excreted through the urine? The answer is glutamine. What is the major source of ammonia excreted in the urine? That is glutamine. Now, comparable to glutamine formation is asparagine formation. Instead of using glutamic, you use aspartic, converting it to asparagine. Same, you need one ammonia to do that. So that's one way to lower also the ammonia level. Urea formation is the major means by which your liver detoxifies ammonia. And among these four ways of detoxifying ammonia, urea formation is the most active. Why? Diba I said, all amino acids are metabolized in the liver except the branched chain amino acids. So because they are metabolized in the liver, that's where you produce the ammonia. So ang daming ammonia na naproproduce sa liver, that's why urea formation is very active in order to detoxify those ammonia resulting from the catabolism of the different amino acids. So the urea cycle, also called Krebs-Hensleit cycle, Hensleit. Same Krebs who discovered the citric acid cycle. Hensleit is a student of Krebs. So you can have your own cycle also. You can discover one cycle. Now, if you look at the pathway, it uses both cytosol and mitochondria. There are only few pathways that uses both. Majority of your pathways either uses cytosol alone or mitochondria alone. Example of pathways utilizing the cytosol alone. Can you think of one pathway that you have studied na sa cytosol lang? All right, glycolysis, very good. Glycolysis, glycogenesis, glycogenolysis, HMP shunt, these are all in the cytosol only. How about mitochondria? Krebs, the Krebs cycle is in the mitochondria. Also your electron transport chain. Now, there is one pathway that you have already studied that uses both cytosol and mitochondria also. That is in relation to your chemistry of respiration. What is that pathway? Synthesis of your porphyrins, heme. Diba? In the synthesis of heme, there are reactions inside the cytosol. There are also reactions in the mitochondria. So that's one pathway that uses also cytosol and mitochondria. Another one is gluconeogenesis. In gluconeogenesis, you are actually using two pathways going reverse. That's glycolysis and the Krebs cycle. The glycolysis part is in the cytosol. The Krebs cycle part is in the mitochondria. So gluconeogenesis also uses both cytosol and mitochondria. And of course, this one, the urea cycle. Now, first reaction inside the mitochondria. 2 moles of ATP, carbon dioxide, ammonia, and water reacts to form carbamyl phosphate. Enzyme for this is carbamyl phosphate synthetase 1. Importante yung 1 because there is a carbamyl phosphate synthetase 2 that you met under pyrimidine metabolism. Differences between CPS1 and CPS2. CPS1 for the urea cycle, CPS2 for pyrimidine. CPS1 is inside the mitochondria. CPS2 is in the cytosol. Third difference, CPS1 requires an activator called N-AGA, AGA, acetyl glutamic acid. That's the activator of CPS1. CPS2 does not need an activator. So those are the differences between CPS1 and CPS2. Second reaction, ornithine plus carbamyl phosphate produces citrulline. Take note, citrulline came from the cytosol. It is not inherent in the mitochondria. It has to enter the mitochondria, and so it requires a transporter. There is a syndrome where the ornithine transporter gene undergoes mutation. So, if it undergoes mutation, no more transporter for the ornithine. It cannot enter anymore your mitochondria. Later, I will tell you that syndrome where your ornithine cannot enter. Third reaction in the urea cycle, citrulline reacts. Citrulline muna gets out into the cytosol. It will now react with one more ammonia through aspartic acid to form arginine succinate. So if you look, there are two ammonias entering. One in the first step, another in the third step. And you can verify that from the formula of urea. Two ammonias are there. So per turn of the urea cycle, you are detoxifying 2 moles of ammonia. Now, fourth reaction, arginine succinate is cleaved to form fumarate and arginine. And then arginine is cleaved further to form urea and ornithine. As I've said, urea represents the final end product of protein metabolism in your body. The more proteins that you eat, the more urea that you excrete in your urine. Now, urea is diuretic. So it will increase the volume of your urine. Urea also is solid, so it will increase the specific gravity of your urine. So what happens if you go on a high protein diet? High protein diet, high urea excretion, high volume of urine, high specific gravity of urine. Reverse, if you go on a low protein diet, low urea excretion, low volume of your urine, low specific gravity. Okay? Now, if you look at number 5, you produce arginine. This is the one that I said a while ago. You can synthesize arginine through the urea cycle. But, as you can see, It is immediately converted to urea and ornithine. It does not stay long as arginine. So if you want arginine to be used for protein synthesis, you must provide it in the diet. The arginine that you would like to use for protein synthesis must come from the diet, not this one, because it is immediately converted to urea and ornithine. So that's the reason why arginine is semi-essential. You synthesize it through the urea cycle, so it's non-essential. But you cannot use it for protein synthesis. You must provide the arginine that you use for protein synthesis in the diet, so it becomes essential. Okay? Now, reaction number 4. You produce fumarate. Fumarate is also an intermediate of the Krebs cycle. So you can actually link the Krebs cycle here. Krebs cycle on the left side, then you have your urea cycle on the left side. The link is fumarate. That's what you call the Krebs bicycle. Bicycle, 2 wheels, urea cycle and the Krebs cycle, with fumarate as the link between the two. How many ATPs do you produce through the urea cycle? Is there any reaction here where you release ATP? Wala. None. So you do not produce ATP, but you use. 2 moles of ATP are used in the first step, 1 ATP used in the third step. Now, usually, if ATP undergoes hydrolysis, the products are ADP plus PI, not AMP plus PPI. Here, you have PPI. To complete the reaction, the PPI must be converted to 2 inorganic phosphates, 2 PIs. That will need 1 more ATP. Convert the PPI to 2 PIs, 1 more ATP. So, how many moles of ATP do you need? 2 plus 1 plus 1 for the PPI. 4 ATPs per turn of the urea cycle are needed. Now, what are the control points of the pathway? Number 1, number 2, number 5. 1 to 5. Carbamyl phosphate synthetase 1, ornithine transcarbamylase, and arginase. They are the control points of the urea cycle. If you want to stimulate the pathway, you stimulate the control enzymes. If you want to inhibit, then you inhibit the control enzymes. Now back to ornithine. I said you need a transporter here. There is a syndrome where the ornithine transporter gene undergoes mutation. So there will be no transporter for ornithine. Therefore, it will not enter the mitochondria. In that case, the urea cycle will stop. No more ornithine. It stops after the first step. And that will now lead to the accumulation of the ammonia. So hyperammonemia will result. The ornithine, because it cannot enter the mitochondria, will also accumulate. So you have a hyperornithinemia. This citrulline gets out into the urine, and you call that homo, not hyper, homocitrulinuria. Urea is urine. Those are the 3H in the triple H syndrome. Hyperammonemia, because ammonia accumulates, because the urea cycle stops. Hyperornithinemia, because ornithine cannot enter the mitochondria. And homocitrulinuria, because citrulline now will be excreted through the urine. The cause of the triple H syndrome, mutation of the ornithine transporter gene. Now, if any one of those enzymes becomes deficient, urea cycle stops and ammonia accumulates. So hyperammonemia will result if any one of the enzymes of the urea cycle becomes deficient and you have the so-called inborn errors. You were born with a defect. Wala ka nung enzyme. So if the carbamyl phosphate synthetase 1 is deficient, the inborn error is called hyperammonemia type 1. If the second enzyme is deficient, hyperammonemia type 2. If the third enzyme is deficient, you have citrullinemia. There will be accumulation of citrulline. And argininosuccinate aciduria, if argininosuccinase is deficient, argininosuccinate goes out into the urine. If arginase is deficient, arginine accumulates, you have arginemia. So as I've said, all of this will be manifested by hyperammonemia, which can lead to ammonia intoxication. So, what kind of diseases can easily give you ito yung triple H syndrome? Hyperornithinemia, hyperammonemia, homocitrulinuria. Mutation of the ornithine transporter gene. All right. So what kind of diseases can easily give you ammonia intoxication? Severe liver diseases. And why is it that you easily go into coma if you have a severe liver disease? Because the urea cycle will stop, because the urea cycle is in the liver. So, if the urea cycle stops, ammonia accumulates in the liver. So many ammonias until such time it spills out of the liver. Puno na yung liver. The ammonia now spills out into the systemic circulation. It will now go to the different parts of the body and finally reaches your brain. And as I've said, the brain is very sensitive to ammonia. So it will try to detoxify the ammonia through glutamine formation. It will react glutamic acid plus ammonia in the presence of the enzyme glutamine synthetase to produce glutamine. Glutamine goes to the kidneys. Once in the kidneys, glutamine is reversed to glutamic acid and ammonia. The ammonia now can get out of the urine because you are now in the kidneys. The enzyme that reverses the glutamine synthetase step is the enzyme glutaminase. So now you can excrete the ammonia through the urine. However, because the liver is diseased and there's too much ammonia reaching the brain, there will come a time, no more glutamic acid. Wala na. So what will the brain do now? The ammonia keeps going to the brain. No more glutamic acid to convert it to glutamine para ma-excrete mo sa kidneys. So what will the brain do? The brain will try to produce glutamic acid. Ubus na to. From where will the brain get glutamic acid? From alpha ketoglutarate. Remember, glutamate dehydrogenase is also present in the brain. So, this enzyme can convert alpha ketoglutarate plus ammonia to glutamic acid. And then, the glutamic acid can combine with ammonia to form glutamine that goes to the kidneys so that you can now excrete the ammonia to the urine. Masaya uli si brain mo. Meron na siyang glutamic acid. Kinuha niya yung alpha ketoglutarate. From where did it get it? From the Krebs. Remember, alpha ketoglutarate is an intermediate of the Krebs cycle. So, what happens to the Krebs? it stops. You are going to deplete the alpha ketoglutarate of the Krebs. And so it stops. And what is the Krebs cycle? That's the major ATP producing pathway in your body. Remember, per turn of the Krebs cycle, you are producing 10 ATPs. And nag-stop siya. So, no more ATP for your brain. And so, you go into coma. See that? So, what is the immediate cause of coma? It is the depletion of the alpha ketoglutarate in the Krebs cycle. Why was it depleted? Because it was converted to glutamic acid by the glutamate dehydrogenase reaction. Na-deplete siya. Kaya wala ka ng ATP production. So, to summarize that, if you are asked to explain the mechanism behind the occurrence of coma whenever you have a severe liver disease. In severe liver disease, the urea cycle stops because the urea cycle is in the liver. Ammonia will accumulate in the liver and will then spill out into the systemic circulation until it reaches the brain. The brain tries to detoxify by converting it to glutamine. Then glutamine goes to the kidneys, and then it will be converted back to glutamic acid and ammonia so that ammonia now can get out through the urine. However, because there's too much ammonia going to the brain, eventually, glutamic acid in the brain will all be used up. No more. So the brain will try to produce glutamic acid. And the best source is alpha ketoglutarate of the Krebs cycle. In the presence of the enzyme glutamate dehydrogenase, alpha ketoglutarate is converted to glutamic acid. So the brain is happy again. It has glutamic acid to combine with ammonia to produce glutamine that can go to the kidneys so that the ammonia is excreted through the urine. However, the Krebs cycle will stop because the source of the alpha ketoglutarate is the Krebs. It will be depleted of alpha ketoglutarate. So when the Krebs stops, no more ATP production in your brain, you go into coma. Clear? That's how to explain the mechanism behind the occurrence of coma whenever you have a severe liver disease. All right. So how do you treat ammonia intoxication? Whenever you treat a disease, whenever you have a patient, whether you are in your clinic or a hospital setting, always tell the patient what kind of diet he is going to take. Do not assume that he knows. Maski regular diet pa yan, you still have to tell him, oh, you can take anything, you can eat anything because there's no restriction. Pero, halimbawa, hypertensive yung pasyente, then you can prescribe a low salt diet. Explain to the patient what a low salt diet is. How low is the sodium na pwede niyang i-take. Pag may allergies naman, you write there, hypoallergenic diet, and then indicate whether to what your patient is allergic to. Para alam nung dietitian kung anong iiwasan. So hypoallergenic diet, patient, parenthesis, patient is allergic to shrimps, crabs, etc. Yun yung allergies ko kasi, shrimps, crabs. So it must be clear what kind of diet you're going to give to your patient. Now, it says there, limit the protein intake. Remember, ammonia comes from proteins. So hindi pwedeng high protein diet. There will be so many ammonias that you're going to form if you give the patient a high protein diet. Pero hindi naman pwede, it cannot be that you do not give protein at all. Hindi naman pwedeng no protein diet. Why? Because you have other important proteins to synthesize. Remember, your hemoglobin, your antibodies, your collagen, your keratin, they are all proteins. Your enzymes, they are all proteins. So how will you synthesize them if you have no protein at all in the diet? So it must be a low protein diet, but give complete protein. Kasi even if it is low in amount, but high in quality, you are sure that all the essential amino acids are there. So low protein diet, but high quality or complete to ensure that the patient is still producing the necessary proteins. Now, you can give lebulus or lactulose to prevent absorption of ammonia in the GI tract. Whatever ammonia goes to your GI tract, you can prevent its absorption by giving lebulus or lactulose, and then you promote the excretion in the stools. Hindi siya na-absorb, so it will be excreted in the stools. You must give antibiotics to kill the normal bacterial flora. In your GI tract, merong bacteria palagi. These are harmless bacteria. That's what you call the normal bacterial flora. Harmless. They do not cause disease. However, they produce ammonia by bacterial fermentation. So you have to kill them also by giving antibiotics. Kill the normal bacterial flora, which also produces ammonia through bacterial fermentation. Sometimes, if you read textbooks, it is specified that the antibiotic that you have to give is neomycin. Kasi mura lang yun. It's cheap. No need to give very expensive antibiotics. Third generation antibiotics, which cost 80 pesos or more per tablet. Tapos 3 times a day mo, 240. And you are just killing a normal bacteria. So no need for that kind of antibiotics. Just give the cheapest one because you're just killing a normal, harmless bacteria. You just want it not to produce ammonia. You can also remove excess ammonia by giving the so-called binding compounds. These are compounds that will bind to the amino acid. Example, remember, for every amino acid, there is an amino group that is attached, which becomes the ammonia. So when you bind the amino acid and you excrete it in the urine, it is just like excreting 1 mole of ammonia per amino acid. So these binding compounds are very important. One binding compound is benzoic acid. It binds with glycine, converting now glycine to hippuric acid. Then this is excreted through the urine. So naka-built-in na yung ammonia doon sa hippuric acid. That's for glycine. Actually, this is a very important detoxifying mechanism in your body. You get benzoic acid from fruits and vegetables. And it is toxic by itself. Yung benzoic, too much of benzoic acid can cause diarrhea. Pero, you do not have diarrhea every time you eat fruits and vegetables because your glycine detoxifies it. It converts it to hippuric acid, which is no longer toxic. So that's a detoxification mechanism in your body that takes place in the liver. You detoxify whatever benzoic acid you have in your diet, convert it to hippuric acid so it's no longer toxic. Another binding compound is phenylacetate. This binds with glutamine. So you will now excrete glutamine as phenylacetylglutamine. So all of this, we can lower the ammonia level. However, it does not treat the cause. Whenever you are dealing with a patient, you have to know the cause of the disease. You do not only treat the symptoms. For example, a patient comes to you with fever. So you give paracetamol for that fever. But you must know also what is causing the fever. Because even if you give paracetamol and you do not know what caused it, hindi siya gagaling. How about if the cause is a UTI, urinary tract infection? So you must give antibiotics and that will be the treatment for the cause. The antibiotics will kill the bacteria causing the urinary tract infection. So you must know the cause. And all of this does not treat the cause. They are just supportive treatment. So what is the treatment for the cause? You replace whatever is missing. In our discussion, what is missing or lacking? Alpha-ketoglutarate. Remember, the immediate cause of the coma is a depletion of the alpha-ketoglutarate. So you must replace it. Replace whatever is missing. Kaso, there is no preparation of alpha-ketoglutarate that you can give. None. So, what do you do in order to prevent the depletion of the alpha-ketoglutarate? We give glutamic acid. Why? You look at this. The reason why you got alpha-ketoglutarate is because you ran out of glutamic acid. So you converted the alpha-ketoglutarate to glutamic acid. And so you depleted the Krebs cycle of alpha-ketoglutarate. If you have an external source of glutamic acid, galing sa labas, there is no need to get the alpha-ketoglutarate of the Krebs. Meron ng pinanggalingan sa labas eh. So you're going to spare the alpha-ketoglutarate from being used up. Your Krebs cycle will not be depleted of alpha-ketoglutarate. It will not stop. So your ATP production goes on and you do not develop coma. So that is the best to give. The glutamic acid. It spares the alpha-ketoglutarate from being used up. Because if you have an external source of glutamic acid, no need to get the alpha-ketoglutarate of the Krebs cycle to produce glutamic acid. Kuha nyo? Any questions so far?
None po, Doc.
Okay. So, that is what happens to the ammonia. The other product of the amination is the keto acid. Ano naman mangyayari sa keto acid? You can have the so-called synthetic pathway where your keto acid reacts again with the ammonia to form back the amino acid. We call that synthetic pathway. So you can react alpha-ketoglutarate plus ammonia, you form back glutamic acid. Oxaloacetate plus ammonia, you form back aspartic acid. Pyruvate plus ammonia, you form back alanine. Glyoxylic acid plus ammonia, you form back glycine. Ganun lang. You also have the so-called glucogenic pathway, where the amino acids enter gluconeogenesis. They are converted back to glucose. A while ago, you have the classification of the amino acids. There is only one amino acid that you cannot convert to glucose, and that is leucine because it is purely ketogenic. Now, as I've said, for your amino acids to become glucose, to enter gluconeogenesis, they must be converted first to keto acids, which are members of the Krebs cycle. You will see how they enter later. They can also be converted to ketone bodies. And as I've said, there is only one amino acid that is purely ketogenic, and that's leucine. You cannot convert it to glucose, only to ketone bodies. There are 5 amino acids that you can convert to glucose and ketone bodies at the same time: isoleucine, phenylalanine, tyrosine, tryptophan, and lysine. Pareho, glucose and ketone bodies. All the rest, they are just purely glycogenic. They can be converted only to glucose, but not to ketone bodies. So this is easy to remember. Only one purely ketogenic, leucine. 5, both glucogenic and ketogenic. And then the rest are just purely glucogenic. Pag hindi kasama sa 5 at saka leucine, purely glycogenic siya. Now, if these amino acids enter gluconeogenesis, the first reaction that they have to undergo is deamination. Deamination means you're going to remove the amino group so that the amino acid becomes a keto acid. Kasi the members of the Krebs cycle are the keto acids. Keto acid lahat yan. So first step, if you want to the amino acids to become glucose, deamination. Then, you must know the entry points in the Krebs cycle. Pyruvate is the entry point of 5 amino acids: alanine, cysteine, glycine, serine, threonine. Lahat yan pwedeng maging pyruvate. Threonine has to be converted first to glycine. Nako-convert kasi yung threonine to glycine and then glycine to pyruvate. So 5. Oxaloacetate is the entry point for aspartic acid and asparagine. Fumarate is the entry point for phenylalanine and tyrosine. Now, succinyl-CoA is the entry point for isoleucine, methionine, and valine. Arginine, glutamine, histidine, and proline can be converted first to glutamic acid. Then, the glutamic acid is converted to alpha-ketoglutarate. So, alpha-ketoglutarate pa rin yung entry point ng 5 na amino acids. For those that are ketogenic, these are the entry points. Ketogenic lang yan. Okay? So, you must remember what are the entry points of this amino acid before they enter gluconeogenesis. Next time, we are going to discuss the individual pathways for the different amino acids. There are 20 of them, so there are 20 metabolic pathways. But you are not going to memorize the pathways. Mabubuang kayo if you do that. I will only require you to know what are the important substances that you derive from each amino acid and what are the important inborn errors that you find in the pathways. So, kanya-kanyang inborn error yan. This is what we're going to take up next meeting. We still have one meeting, di ba? For protein metab. Okay. Do you have any questions? Wala naman?
Apo.
Okay. If you don't have, sa bagay, 2:30 lang. You can stop now. Kaya lang do not log out. Kasi you're